Hello everyone, my name is Nima Madani, and I just wanted to take the time to talk to you about a project that I did for Dr. Holly's class. This is for PhD 6937, Environmental Ecology of Human Pathogens, and the topic I chose is monitoring cholera in Haiti water reservoirs. Before we begin the presentation, I just wanted to lay out a quick outline of some of the topics I'll be hitting while discussing this presentation. The first topic I wanted to touch on is the history of Vibrio cholera. Followed by that, I want to go into the background of kind of what's going on with the Haiti situation. Then I'm going to explain more about the persister phenotype. After that, we'll hit into some methods and data for the study that was done. And then to wrap it up, we're going to look into the public health implications, just kind of give you a little reason why the work that is being done with this study is very important to the people and how it affects even us here in the United States. All right, to begin the presentation, I just want to start off with a little bit of background on what is Vibrio cholera. Vibrio cholera is a small bug that's part of the cholera pathogen that infects the small intestine. It is a gram-negative pathogen that is in the shape of a comma and has one flagella. The flagella is its motive it uses for transportation within a cell. This Vibrio cholera uses a CTX phage to infect humans, and how the phage infects the humans is that it uses a TCP, which is a special kind of pilus that it can use to attach to the small intestine cells of the human and then secrete in a special CTX toxin. Once that toxin goes into the small intestine, it causes it to flush out all of the water and the electrolytes that the cells use to communicate, such as sodium ions and potassium ions. By flushing out the water and the sodium ions, these cells aren't able to, the human small intestine cells aren't able to communicate with each other. And by not being able to communicate, this causes a huge dilemma as that water passes through without it to absorb the nutrients. This, of course, causes diarrhea, and in severe cases, it can lead to death. There have been 7 known pandemic waves of Vibrio cholera, which I'm about to go into. On here, I have 2 figures to help explain kind of the 7 different ways of the pandemic that happened with cholera. The one on the top figure is just kind of like a timeline to explain that during the 7 ways how long each impact was. As you notice that if you look at just the first 5 pandemics, they all take place in the 19th century, and they all kind of have a duration from about 8 to about 23 years, which isn't anything major. The 6th pandemic takes place heading into the beginning of the 20th century, and then after that 6th pandemic, you notice that there's about a 30-year lull in between before we get hit with the 7th pandemic. For my presentation, I just want to focus on the 7th pandemic, the El Tor strain, because that is the most recent one to hit the Haiti region. Now I would like to begin with the background of what was going on in Haiti before cholera struck. So about 2010, there was an earthquake that devastated the Haitian region. That earthquake had a magnitude of 7.0, which on the Richter scale, it can be pretty logarithmic, so it was pretty bad. And Haiti is already a third world country, one of the poorest countries in the whole world. So a lot of efforts had to come to help it. And the United Nations decided to get involved and send a couple of peacekeepers, which is very common what it was. What unfortunately happened was a couple of Nepalese peacekeepers were carrying an asymptomatic strain of the Vibrio cholera on them when they were doing peacekeeping work. Now, keep in mind, in 2010, Haiti hadn't had any exposure to the cholera strain for the past 100 years. So by having no outbreaks in the past, this population was very susceptible to the pathogenic response of the cholera. So when they first initially got attacked, it was an Ogawa strain, which is kind of how they classify the different stereotypes, which I'll go more into later. But over time, through 2017, it slowly started to transition to become more of an Ogawa strain. If you can focus your attention on the figures presented by the WHO, as you can see, beginning early on in the 2010, you can see that there was a high increase in cases, suspected cases, and hospitalizations. You also see that there was a high amount of death in the beginning, but as we go through time, all the way until 2017, it starts to dwindle down to about 0. Now, many scientists are working on this to figure out to see, has the cholera maybe run out of nutrients and it's unable to fit to survive in the environment since it hasn't been there before, or has it found a way to persist and then is looking for another day to strike? Now, as I alluded to earlier about the question of whether the cholera strain is persisting in the environment, some of the research I've been showing, especially some magnificent work done by Dr. Afsar Ali, shows that there is evidence of a persister phenotype. To go more into detail, I just wanted to point out the figure on the top, how it has 3 phases: a formation, maintenance, and exit phase. Now, to get a little bit of the color coding, you can see the normal cells are green, not culturable but non-viable cells are in light red, and then the dead cells are in red. Keep a quick attention to the black cells, which are your persister types. So what happens is usually a harsh environment, lack of nutrition, or an antibiotic is introduced and kills about 90% of the culture. But fortunately, they have these persister cells, which are very resilient and use very low energy. They can survive the harsh conditions, and then once the harsh conditions have passed, they can once again activate and prosper to help grow. A great analogy for this is the TV show The Lone Survivor. In this show, the president gives a State of the Union address and he takes his whole cabinet. Usually they keep one cabinet member, though, in a separate room, separate from the State of the Union, in the event that if something were to happen, then the one cabinet member can take over and take control while they recover from it. In the event of the show, unfortunately, a bomb strikes and takes out the rest of the cabinet, making the one cabinet member the lone survivor, and now he has to go on and carry on with the country. This can kind of be seen with the persister cell too. The persister is the one that has to survive the harsh conditions, and then once everything goes back to normal, they can able to thrive again to help remultiply the colony and survive. So to test for the persister phenotype, I'm just going to give a quick review of the methods and protocols that we did. That way we can see a little bit more into our experimental question. The first thing we had to do was make sure we were isolating the correct O1 and Vibrio cholera strains. So what we used was peptone water, or APW, which was an enrichment technique just to make sure that the cholera was thriving and not other pathogens. We were able to streak that enriched plate and put it on an LCBS agar plate and incubate overnight at 37 degrees Celsius. Once we were able to do that, we grabbed 6 to 8 special colonies from the LCBS plate and streaked it again on agar plate to incubate and observe the growth at 37 degrees overnight, again, same protocol. Once we were able to grow it on that plate, we performed a process called agglutination, which targets a specific polyvalent O1 antiserum. Now, the reason agglutination works is that you can identify different types of Vibrio cholera by their serology, or the specific types of antigens that it has on the cell. How agglutination works is that you get special antibodies that are only specific to the O1 serum, and these specific antibodies bind to the antigens of an O1 cholera cell. So then a positive marker will give off, letting you know, hey, we have detected some O1 antiserum. Once we were able to confirm that that specific sample was an O1 strain, we then went again to double check with performing PCR, or polymerase chain reaction. So to begin with the results, the first one I want to show is the table on the right, table 1, which is the effects of diverse enrichment conditions with the isolation of culturable Vibrio cholera O1 strains when we isolated them from the Gressier and Gresca regions of Haiti. The main takeaway message from this is that on the top right corner, which I'm going to circle with the pen here, we proposed 3 different conditions for culturing our cells. When we were able to culture them, we saw that in the first condition, incubating at 37 degrees for 6 to 8 hours, we were able to observe 3 of the 5 O1 serums. We decided to switch the conditions the second time to 37 degrees overnight, and we only observed 1 of the 5 pathogens. But the big change is when we changed it to 40 degrees at 6 to 8 hours, we observed 2 pathogens that we hadn't seen before. The main takeaway from this graph is that it's always important to try new methods when you're incubating, because you can always discover new strains, and that can be one step closer to finding a potential reservoir. The second table is the one that shows the PCR to confirm to make sure that when we were isolating these O1 genes, we were getting the ones that actually are confirmed to be O1 Vibrio cholera, what we were looking for. The first two that I'm circling right now are just genes to confirm the genus and the species. ToxR is something that's very specific to the Vibrio side of all cholera, and OMPW is very specific to the cholerae, which is the genus and species of how it's identified. As you can see here, there is no any marker for the CTXA, CTXB, or any of the pili genes except for the bottom one. For O1 Vibrio cholera to be toxigenic, it usually has these two parts and components to it, but it's puzzling here to see that it's not in there. All right, and here's the main table where all of the work paid off. Right here, we were showing that when you look at the polyvalent antiserum, we were able to detect that, hey, we found an O1 strain here. Now, the main two strains that I previously mentioned that we found in Haiti were the Algawa and Naba antiserum. Algawa was originally the first strain that came to Haiti, and over time it transitioned to becoming a Naba. But if you take a look at the table here, only 2 out of the 5 are actually following the Algawa or Naba antiserum trend. The ones that I'm circling on the left, the first, second, and fourth one, are actually strains that have no identified antiserum that we've seen to this day. This shows that if there's a different antiserum being developing, that is developing different genes to better persist into the environment. By being able to see this, we need to do more testing to confirm what kind of antiserum it is, and maybe we can discover potentially new ways of seeing, okay, is this surviving in the environment, what genes is it doing, and has it found a potential reservoir to best suit its needs? Now here are just some conclusions and implications to better summarize the importance of why this study was found so impactful. First one is, of course, vaccine development. Now, whenever you're making a vaccine, you usually want to target specific antigens of the pathogen of interest, and that way you can develop certain antibodies so your body can develop an innate immune resistance. That way, when it's exposed in a greater dose later, I'll have a ready response for it. If you're seeing that you're developing different from the Algawa and the Naba antiserum, then you make a vaccine just for those two, and the environment creates a new strain. Of course, the vaccine's not going to be very effective, and the population's still going to get sick from cholera, making your vaccine pretty useless, which is definitely not good because vaccines can get pretty expensive. The second big implication of the study shows how the variation of incubation times has increased the isolation frequency. What that basically means is that when we tried our first method at 37 degrees, we were able to isolate 3 out of the 5 strains. But had we not gone to the 40-degree incubation, we would have missed out 2 completely different strains. Those different strains could be carrying different types of antibodies, antigens, or just cause a different impact with a whole new mechanism or a whole new strain. Once again, to go back to the vaccine development, if you're not detecting all of the specific cholera cells in the environment, and you make a vaccine to target all of them, you're going to run into the same dilemma again as I talked about earlier. The third big trend is that we got one step closer to identifying environmental rules that can trigger the persister cells or the VB and C cells from going to dormant to awake. Of course, this is a big trend that everyone's trying to figure out when they are studying Vibrio cholera, is what causes it to become active and infect humans. By looking at this and figuring out the trends in the environment, this potentially gets us one step closer to an environmental reservoir. If we can find the conditions that cause it to become awake or cause it to be dormant, we can then target specific sites in the environment that we figured will carry these conditions to allow it to persist. Also, if we can figure out what nutrients it needs to become active and what nutrients it lacks to make it become dormant, we can better cultivate the environment to make sure it doesn't get that key nutrients. So even though that cholera is in the environment, it stays dormant, so it's not really a major threat to humans. And finally here, I just have a couple of references. Please feel free to reference any of these 4 sources so that way you can get more information on the topic if you're definitely interested. I also just want to take the time to thank Dr. Ali and his student Mira Alom for exposing me to this method and definitely broadening my educational experience. Thank you, everyone, and I wish you a great rest of your finals week.